We're going to talk about microbial genetics. And here, genetics is a study of inheritable traits and how they're expressed. The genome refers to the entire genetic complement that's on the DNA in the organism, so it's going to include the genes, all the nucleotide sequences. The structure of the nucleic acids, you have a sugar, you have a phosphate group, and then you have a nitrogenous pair. Now, with the nitrogenous bases, certainly with DNA where you have two strands of it, the nitrogenous bases will pair up. One from one strand pairs with a base from the complementary strand across from it. And in this diagram, it is showing the structure of the bases. So in A, you have adenine and thymine, A and T, that bind together by hydrogen bonds. That is the red dotted line. As you can see, there are two of them. In figure B, you can have adenine hydrogen bond, once again by two bonds, to uracil. This would be found in RNA. Uracil is found only in RNA, thymine is found only in DNA. And then both RNA and DNA have the GC complex, the guanine and cytosine, that will bond together by three hydrogen bonds. So this is just the base component of the nucleic acid. So with the nucleic acid, those were the bases as seen. They look like the colored strands sticking off of the backbone of, in this case, DNA. The backbone is alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, and the base is connected to the sugar. As you can see, because you have DNA is usually double-stranded, when you put the two strands together, the base from one strand is pointing inward and hydrogen bonds to the base from the adjacent strand. When you look at prokaryotic chromosomes, number one, they're circular. Prokaryotic cells are haploid. That means they only have a single copy of the chromosomes. They don't have two copies. And you're looking at DNA as far as containing that genetic information. In this picture, on A, you see the represent drawing of the nucleoid region is where the DNA is in the cell. Remember, there's no defined nucleus. In B, what you're looking at is the bacterium as it is lysing, and you see all the strands of the chromosome, which is the DNA. There's a small little piece down at the bottom that is labeled as a plasmid. That is extra small circular piece of DNA that is in the cell. Plasmids will replicate independently of the DNA of the bacteria. It's not necessary for required for survival. It may be there, they may not be there. Sometimes they give, if they are present, they contain genes that will give some advantages for survival for the bacteria. So there's different types of plasmids. There are some that give fertility factors. From a medical standpoint, one huge problem are the resistance factors. They often contain genes that allow for resistance to antibiotics, so that is a concern. And some of them often have virulence plasmids, which means it increases the virulence or pathogenicity of the organism. For eukaryotic cells, the DNA is in the nucleus, so that's where the chromosomes are going to be found. There's usually more than one chromosome. Most of cells are diploid, meaning they have two copies of everything. The DNA is also linear. It is not circular. So the way that in a eukaryotic cell, the way the DNA is going to be packaged, you have that DNA. It's going to be coiled and wrapped around certain proteins that are known as histones. Then you have several of these complexes that are stacked together. It's very, very tightly coiled up. It's highly condensed, especially when it is going through cell division, through mitosis or meiosis. In eukaryotic cells, there can be some extra nuclear chromosomes, DNA, extra DNA that is found in the mitochondria and in the chloroplast. The DNA that is found there is circular, so it resembles more closely to prokaryotic DNA. Some fungi and some protozoans may also have platelets. So this table is showing a comparison of the bacteria versus archaea versus the eukarya, the three different domains, and some characteristics of their genomics, of DNA characteristics. First thing we're going to look at is DNA replication. How does the DNA make a copy of itself? So sometimes it's referred to as DNA synthesis or DNA duplication. No matter what you call it, is how does it make an extra copy of itself so that after replication or duplication of the cell, after cell division occurs, each brand new cell will have its own copy of DNA. So this obviously has to occur prior to cell division. The replication is referred to as semi-conservative. What that means is that the new DNA, after you've made a copy of it, the two original strands do not go back together. You end up with one parental or original strand and then one new daughter new strand together. So it's not the two original and two brand new. You get one old with one new. And this diagram is just showing that idea of the semi-conservative model, that if the original one is purple, when it's going to separate, so each strand can be used as a template to make new strands, which would be that darker green turquoise color, and that it's going to stay with that original parent that it used as a template. So that original strand, you can see, is going to continue on and remain. When you look at the function of DNA, you're going to see that there are different things that are required. You have the existing strand. You're going to add the nucleotide one at a time to it. It's actually going to be a triphosphate nucleotide. You add it on, and then two of the phosphate groups are going to be released. So you're going to basically add on one base at a time, one nucleotide, which has the base, the sugar, and the phosphate group, one at a time. The process for replicating the DNA follows a very tightly controlled process. It's going to begin in bacterial DNA. It starts at a point of origin of replication. For prokaryotic cells, there's only going to be one origin point. It's kind of like a red flag. Start here, start here. It's going to involve several different enzymes. We're not going to get into all of them. I'm going to kind of summarize it for you. One major enzyme is going to be DNA polymerase. That is going to be necessary for DNA replication to occur. Now, something to realize about this particular enzyme is that it can only read in one direction. It can only read in the 5' to 3' direction. What we are talking about, because I realize a lot of you may not have had chemistry. In chemistry, when we have a compound, we number the carbons on them. So when you look at the sugar in DNA, the deoxyribose sugar, it has 5 carbons, and we number them 1 through 5. And that helps us to understand the orientation as it binds to the phosphate group, binds to the nitrogenous base. In this particular case, the number 1 carbon is always going to be bound to the nitrogenous base. It helps to orient when we number them, like I say, to know exactly what we're talking about. And in terms of enzymes, they can recognize differences depending on what is attached to the various carbons. So the DNA polymerase can only read one direction. The two strands of DNA are what we call antiparallel, meaning they are running in opposite directions. So if you were to start at one end of the very end of the DNA and you look at it, then you would say, okay, one end is the 5' end, one is the 3' end. Because DNA can only read in one direction, then that means, and the strands are antiparallel, one strand of DNA, when you use it as a template, the 5' to 3' strand, the resulting brand new strand will be made in a continuous one long strand. But the other strand, which also is going to be used as a template, you're going to have to unwind a section of it, and it's kind of like the DNA polymerase can only read a small section because it can only read one way. And essentially, it's going to be called a lagging strand. So we're going to see how you make a piece, then you unwind some more, you make another little piece, unwind some more, make another little piece, and then you have to connect all those little pieces together. That's what happens on the lagging strand. So this is showing the initial process. The DNA is double strand, as we just said. It's in a double helix. It's going to involve multiple enzymes. One of the first things that comes in is a DNA helicase. It has to untwist or unwind that DNA. You have other enzymes that come in that have to help relax it. Single-stranded DNA is fairly susceptible to breakdown, and so the two strands are wanting to wind back up, and you have stabilizing proteins that are going to come in and bind to the single-stranded portion. At the point where the DNA helicase is unwinding, untwisting that DNA, we call that the replication fork. Once it has been unwound, DNA polymerase, and there's a DNA polymerase 1 and a DNA polymerase 2. DNA polymerase, as I said, can only read in one direction. And so it can only read in the 5' to 3' direction. So one of those strands will be made continuously, one discontinuously. But in the initial process, you unwind the DNA with the helicase. There's a lot of stress there. With unwinding it, you put in those stabilizing proteins. DNA polymerase comes in, and it's going to help stabilize it. Now, in figure B, the upper one, if we look at the leading strand first, this is the one that's made continuously. First, you have a primer that comes in because the DNA polymerase can't start solely from scratch. It has to have at least one base there to start the brand new strand. So that's what the RNA primer does. This primase comes in, it adds the first couple of bases, and then the DNA polymerase can come in. It's reading that original parental strand, using it as a template. So remember with complementary base pairing rule that you've already covered is that wherever there's a G, it's going to put a C. Wherever there's an A, it's going to put a T. Those bases always pair together. So that's why it can just read along what the existing bases are on the parental strand, and it's adding the appropriate complementary strand. As you can see, it's just reading along continuously, adding in one base, new base at a time. It's going to be just zipping along. In this case, it is moving from right to left. That DNA is going to be continuously unwound by the helicase, and so it continues to open up moving towards the left, and that leading strand is just zipping right along following it. On the lower picture on C, showing the synthesis of the lagging strand, excuse me. Once again, DNA polymerase, you have to have the primer to start because it cannot add just by itself. It has to have at least a couple bases there to get started. So you have your RNA primer there to start. It's going to read a section. Now it's reading from left to right. So what happens here is that initially it started way over on the right side. The primer was there, and then the DNA polymerase came in. It added the complementary bases, just like it does on the leading strand. And then it hits a point where it stops. And then you've opened up a new section. So now new section is exposed. So it would have made the first initial section way back on the right side. And then about where you see the number 7 and you see the red primer, it came in, the RNA primer came in bound there. The DNA polymerase made the next section. Meanwhile, the DNA, the original strand, that helicase is unwinding as we're moving towards the left. So it's opened up another section. You can have more than one RNA primer in the area. So when it finishes, section it gets released, it comes back. Now it has bound over where 6 is, because that section is open. RNA primer bound. As you can see, DNA polymerase came in and bound. Again, it's putting in the appropriate complementary bases at number 8. And it's going to continue as it moves and finishes in that extra little space. Well, you have all these little fragments that are known as the Okazaki fragments. DNA ligase, as shown in number 10, is an enzyme that's going to come in and it's going to connect all those Okazaki fragments together. Kind of think of it like DNA superglue. So it connects all those little pieces together. When it finishes where it currently is shown, it will then move to the section by number 9 where there's that little gap there. That DNA polymerase 1 will remove itself. The DNA ligase will then go there. When it is done connecting those two fragments, the fragment that started at 6 should be completed all the way towards that primer near 7, and then the DNA ligase will connect those together. Because of the extra steps, it takes just a little bit more time to complete this process, so that's a lagging strand. DNA polymerase, because it is circular, it's bidirectional, meaning it starts from that origin and it will move both to the right and left from there. DNA is methylated, meaning you put a methane group on the end. It helps to control the genetic expression. It helps to protect it and is involved with repair. So in this diagram, you see the circular DNA of the prokaryotic cell. You see up the basically the 12 o'clock position, the origin. There's usually only one origin site for prokaryotic cells. Replication, as I said, is bidirectional, so that means it's going both, as you can see to the right and the left. So you're going to have 2 replication forks, one on each side. You can see the original darker parental strand and the lighter strand is the new daughter strand being formed. It just continues and proceeds in both directions until you reach that termination point, and then the 2 coils will separate, and you now have 2 copies of the original DNA. It's identical to what you started with. You have one, for each set, you have one original parental strand and one new daughter strand. Now, a lot of things with eukaryotic DNA replication will be very similar. There are, however, some differences. Remember that eukaryotic DNA is linear, and so you can have thousands of replication origins, not just one. And the Okazaki fragments tend to be a little bit smaller, and there are additional DNA polymerases involved. Now right here I want to distinguish between genotype and phenotype. Genotype, that refers to the set of genes that are on the DNA, that's on that genome. Phenotype is what the physical features that you will see. What do you physically see? And that's going to be determined. What you see is determined by the genes. So phenotype is determined by genotype. So how do you get information? If all the genetic information is on the DNA, how do you get that off the DNA to make proteins? Transcription is the process where DNA is going to be used as a template to make RNA. And then translation is going to use RNA as a template to make proteins. So the central dogma of genetics refers to DNA is transcribed to RNA, and then RNA is going to be translated into the proteins. So as you can just see in this diagram here, DNA, which is a nucleic acid that contains the genotype, all the information for controlling everything in the cell. Through the process of transcription, you're going to use DNA as a template to make RNA. And then through the process of translation, you will use the mRNA as a template to make your proteins. And then it's the presence or absence of proteins that determines the phenotype, which you actually see. Now, remember that both DNA and RNA are nucleic acids. So their building blocks are those nucleotides. You've got those nitrogenous bases and the sugar and the phosphates. But a protein is the monomer building block is this amino acid. So you have this long chain of amino acids. So just keep in mind as we go through this that when we hit translation, it's like we're having to change languages. And I know oftentimes people get transcription and translation confused, and maybe this will help you. When you transcribe something, you're simply copying it. Hi, how are you? You transfer and copy it over to hi, how are you? It's the same language. You're just copying it. And that's what happens in transcription. You are using DNA as a template and you just copy it over to form RNA. When you translate something, excuse me, what you are doing is you are converting from one language to another. And in essence, that's what you're doing here. You are converting from the language of nucleic acids to the language of proteins. So in transcription, you said, hi, how are you? In translation, instead, now you're switching languages. So you're going to have to say, oh, bonjour, comment allez-vous? You're switching languages. You have to deal with that. You have to have a way of handling that. You have to have your translation book, if you will. So we're going to go through this. We're going to go through both processes. And you can see in this picture is just kind of showing how the long red strand is the mRNA. You're going to have your ribosomes, which are the structures of the site where protein synthesis occur. And you have these growing peptide strands. Peptide is a small protein, which are the light-colored balls. Each ball represents an amino acid. So you can see on one strand of mRNA, number one, you can have multiple ribosomes attached. So you can be making a lot of protein at once. For transcription and translation, it's going to involve different types of RNA. We're going to focus on just certain ones of these. For prokaryotes, transcription using DNA as a template to make RNA, that is going to occur obviously in the cytoplasm because that's all it has, but it's going to be in the nucleoid region where that DNA is, which makes sense because you have to use the DNA as a template. There are three basic steps: initiation, elongation, and termination involved with transcription. So what's going to happen for transcription is that on the DNA, there's regions referred to as the promoter site. It's areas that are going to attract RNA polymerase, not DNA polymerase, RNA polymerase. It's similar to DNA polymerase, but it is different. It's involved with transcription. And the RNA polymerase will attach to the promoter area. The promoter area is going to basically kind of form a bubble, and it's going to orient that RNA polymerase as to which direction it needs to read that DNA. It's going, okay, we need you to turn in a certain direction. Is it going to read the entire DNA strand? No, it is only going to read a small section of it. So that's why you have several promoters and it knows where to attach to. So once that is initiation, is the binding of the RNA polymerase to the promoter site. Elongation excuse me, begins when that RNA polymerase starts moving down using the DNA strand as a template to make a new RNA strand. It's using that complementary base pairing rule once again. So wherever it sees a G, it's going to put a C. Wherever it sees a C, it puts a T. Wherever it sees a T, it's going to put an A. Wherever it sees an A, it will put a U for uracil. Because remember, uracil's in RNA, not thymine. So it's going to be just zipping along and reading. This is elongation, so it's adding one at a base at a time. And it's only reading one of the strands. So this shows as it moves along, you have your growing. The red would be the mRNA strand versus messenger RNA that's being formed. The DNA, after the RNA polymerase passes by, the DNA will coil up and close up behind it. So this will continue on until termination occurs, which is when it reaches a termination site or a terminator region, and then it stops. The RNA polymerase will disassociate, break off from the DNA. The RNA strand is released. The DNA is now completely closed back up again. Now, one thing I did not mention during DNA replication, that as the DNA polymerase is moving along, it will often act as a proofreader, trying to guarantee that it is making it correctly. So in prokaryotic cells, that's pretty much all there is to transcription. Initiation, elongation, and then finally termination. In eukaryotic cells, there are some differences. Number one, RNA transcription is going to occur in the nucleus because that's where the DNA is. And there are some additional things that also have to occur. You will still have the initiation, the elongation, and termination, but there are additional steps that have to occur before you end up with that mature RNA strand, before translation can occur. And this is the additional steps are capping, polyadenylation, and splicing. And so what's going to happen is up at the very top, you have your template of DNA with the green and the purple components in it. During transcription, you read through everything. Now, the capping is you're going to put usually a guanine on one end, and then you put a lot of adenines on the tail. So you add the cap and the tail. That's going to help protect the RNA when it has to move out into the cytoplasm. Like I say, you see this in eukaryotic but not prokaryotic cells. Splicing. This occurs only in eukaryotic cells. There is a lot of extra material on the DNA, and that's referred to as introns. The introns are these segments. It's extra material, and it's not needed. But when transcription occurs, as the RNA polymerase is using that DNA template, it's just going to read everything. It doesn't jump and skip over the non-coding parts. It reads everything. So you have those extra pieces on that mRNA strand. So after you put the cap and tail on, you need to remove those extra pieces. So you need to cut out those introns. The exons that remain need to be spliced together or pieced together. And that's exactly what splicing is. You remove those non-essential pieces, leaving just what you need. So the final mRNA is going to be much shorter than the original one. Now, just so you know, if you're wondering why do you have these introns, there is an advantage to having it. In this particular diagram, it's showing exon 1, 2, 3, and 4 that's needed for this making this particular protein. Well, maybe another protein, you might get a message, and in that case, you would need exon 1, intron 3, and exon 4. So it increases greatly within one area the number of different possible proteins that could be made. So once you put the cap and tail on and you've spliced out those introns, now the mRNA is going to move out into the cytoplasm, which is where translation occurs for eukaryotic cells. With translation, this is a process where you're going to now use that RNA as a template to make your proteins. And this is going to occur on the ribosomes. As I said earlier, you have to change languages. You have to now switch from nucleotides to amino acids. How is this going to work? This is what we refer to as the genetic code. This is universal amongst all living organisms, whether you were talking about a bacteria, a fungus, a plant, an animal, humans, whatever. It is the same. This is like your translation dictionary. Every 3 bases is called a codon. 1 codon is equal to 1 amino acid. So in other words, every 3 bases equals 1 amino acid. And this is showing you what those sequence of bases, what those codons are, and for what amino acid they code for. You will notice that you have 4 different bases. The way you pair them up gives you a possibility of 64 codons. One of those is a start codon. It also codes for methionine, but it's the start one. 3 of them do not code for amino acids, and they are the stop codons. One thing that a lot of books do not, in an introductory course, don't mention is what is known as a wobble theory. If you notice, not for all of them, but for several of the amino acids, the first and second base are critical for determining what amino acid it is, but that third position, it may not matter. Whether it's a adenine or whether it's a guanine, you will still end up with the same amino acid. And that's known as a wobble effect, that basically if you were to have a mutation, the mutation was in that third position where you put the wrong base in, it might not make a difference. You might still end up with the same amino acid. So it's not as critical. When we talk about amino acids, you'll hear me say location is critical. Sounds like real estate. Location, location, location. But it's true. That third position is not quite as critical as the first and second by any means. You'll also notice there's a lot of redundancy because there's only, most books will tell you 20 amino acids. There's this cysteine, which is a slight modification of cysteine, which is an amino acid. But basically, if you have 64 codons and 20 amino acids, there's obviously going to be some redundancy there. There are going to be different players with translation. You've got messenger RNA or mRNA. You've got transfer or tRNA. And then you've got the ribosomes, and you also have ribosomal or rRNA. So a single prokaryotic mRNA can code actually for several different proteins, essentially, once again, depending on where you start, where you stop. The transfer RNA kind of looks like this cloverleaf structure is how it's usually designed or described as. RNA is usually single-stranded. If you look in A, at the end, the uppermost area, that is where the amino acid is going to bind on that acceptor stem. That is the amino acid binding site. The opposite end from that is the anticodon region. That's the area that's going to bind to the mRNA during the process. The ribosomal structures, it's made up of two subunits. They're usually separated, and they will come together to form a larger unit for protein synthesis. When protein synthesis is complete, then the two subunits will dissociate again. In figure A, you can see there's a 50S and a 30S that come together to form a 70S. Yes, I know it does not add up. Uh, it's the way they do these certain units, but it is a 70S. That is the prokaryotic ribosome. In B, you've got the 60S and 40S subunits that come together to form the 80S ribosome, which is found in eukaryotic cells. As you can see, it's slightly larger. The ribosomes are going to, subunits come together with the mRNA is going to basically, it kind of like feeds between them. It's helping, the ribosome is, we always say it's the site of protein synthesis. What it's doing is allowing to bring all the players in close proximity to each other so that you can form the protein. You can't form it when everybody's floating around in the cytosol and it's like, get back over here, and no, don't go yet. And it holds everything together in the proper orientation. It's going to feed that mRNA strand through, which is carrying that information from the DNA. On the large subunit, there are certain binding sites. There's an E, a P, and an A site. That's where the tRNA is going to come in. And notice the mRNA is down towards the bottom of it. Well, the tRNA has that anticodon region that binds to the mRNA, and it's going to be held in one of those three sites on the ribosome. And what it's doing is you're making a protein, which is made up of a strand of amino acids. So you've got to bring these tRNAs close enough together that have amino acids attached to them to form the bonds between them. So once again, if we look at the stages of translation, there's three main stages: initiation, elongation, and termination again. And so if we look at initiation, that is, as the name implies, the beginning steps. The mRNA is going to bind to the small subunit of the ribosome. The very first tRNA, which is always going to be carrying the amino acid methionine, comes in. Now it is showing in the first figure the base sequence. So the start codon, remember, was AUG. The anticodon has the complementary base. If the bases do not match up with the complementary proper base, it's just going to bounce off. In this case, it is the correct one. So remember, A and U and G and C bind together. So the anticodon region binds to the codons, and it sticks in place there. Now, the other end of the tRNA, remember, has already picked up the amino acids. Where did it get it from? They're floating around in the cytosol. So it's already picked it up. Once the tRNA binds to the mRNA, then the large subunit comes in to place and attaches, and it has placed, it moves in, so it's placed that tRNA into the P site. That is initiation. It's like, okay, now we're set, we're ready to go. So tRNA comes in, binds at the stop codon, then the larger subunit comes in and binds, puts it to the P site. Now you're ready for the elongation to occur. What happens in elongation is another tRNA is going to come in. Once again, the anticodon on the tRNA has to be complementary to the codon on the mRNA. It has to be complementary bases. If not, it won't bind. It'll just bounce off. In this case, once again, it bound. It's going to be placed into the A site. What this is doing is bringing the amino acids on those two tRNAs. They're sticking off on the other end. You see the MET, which is methionine, and then you see the phenylalanine, which is abbreviated PHE. So 1 and 2, it's brought those close enough together where you form a peptide bond between those two amino acids. This is the whole purpose of, is bringing those players. You've got to make a chain of amino acids, which means you've got to bring two amino acids close enough together to form the bond. That's the purpose of the P and the A site, is holding those tRNAs close enough together so you can form that bond. How do you know what order? That information's on the mRNA. Now, anytime you have binding occurring, I'm always saying there's a shape change, right? Well, yes, there was. So now what happens is everything shifts a little bit. So the tRNA that was on the P site, it shifts over to the E, A shifts to P, and with that shift changing, the amino acid that was bound to that first tRNA is released. What does it do? It goes out in the cytoplasm. Now, tRNAs are very specific. If it picks up methionine, that's the only amino acid it can bind. So it's going to go find another methionine. So now you have the P site filled, but the A was empty because A went to P. So now in comes your third amino acid into that empty A site. It binds, you form a peptide bond. That causes a shape change. So the tRNA in P gets kicked out. A shifts to P. A is empty. A gets filled, form a peptide bond. P gets kicked out. A goes to P. A is empty. In comes the tRNA, forms a peptide bond. P gets kicked out. A goes to P. A is empty. In comes another tRNA, form the peptide bond. P gets kicked out. A shifts to P. A is empty. In comes a new one. And that just keeps going on and on. One by one, you are adding one amino acid at a time. That's what a protein is. This will continue until it reaches a termination site, for which there is basically a stop codon, and there's no amino acid. So nothing's going to bind there, and everything gets released. Your protein is released, your mRNA is released, the two ribosomal subunits will break apart. Now, in prokaryotes especially, you often have what are known as polyribosomes or polysomes. This is where you can have one strand of mRNA, and you can have multiple ribosomes attached to that mRNA, various stages of translation occurring. Because with prokaryotic cells, you do not have a nucleus, and transcription and translation are both occurring in the cytoplasm. You can theoretically be having transcription and translation occurring at the same time. So you can be making that mRNA, and before you've even finished making the mRNA, you can start translation. So like I said, termination is when you reach that stop codon, and everything dissociates. For eukaryotes, there's a little bit of difference with some of the practices of translation. In general, the practice is very similar for purposes of this. Know those general steps. So this is a table that is comparing replication, which is the process of making another copy of the DNA, transcription, which is using DNA as a template to make RNA, and then translation, which is using RNA as a template to make proteins. So how do you regulate what we call genetic expression? How do you regulate the making of the various proteins that these genes code for? Some of them may be what we say expressed all the time, meaning they're turned on all the time. But other ones will only be turned on, the genes will only be turned on when you need that product. It's a way of conserving energy. Why make something if you don't need it? An operon is an area on the DNA. It will consist of usually a group of genes that are similar for making something, and the promoter area. So that's what we call an operon. And it's going to be controlled by an area called the operator. You can control maybe several genes as a single unit, and it makes it easier to turn on and off very quickly. So here is an example of kind of a classic operon where on this DNA strand you can see you may have several genes. In this case it's showing 4 different genes. Instead of controlling each one of those genes separately, it's controlled as a single unit. So you're going to have a promoter area, you're going to have an operator area. The promoter, the operator, and the genes collectively are known as the operon. Now, nearby, what we call upstream from that, there's going to be usually some regulatory genes. Some of these operons are inducible ones, some of them are repressed. So there's different ways that they can be operated. The 2 most well-studied operons are the lactose and the tryptophan operon. And so we're going to be looking at those. This is showing with the lactose operon the direction in which you're going to see the transcription occur. So let's look at this in a little bit more detail. In the upper picture of the lac operon, number 1, you've got 3 genes that control the production of the 3 proteins that are necessary for breaking down lactose, which is milk sugar. So like I say, the purpose of an operon is going to control it as a single unit. You've got that operator region and then the promoter area. Now, you have above on there, it's not part of the lac operon, but it's right near it, is what we call the promoter and regulatory gene. That is always turned on, and you have transcription and translation occurring there for making a repressor. So the repressor is always being made. When lactose is not present in the media, why make the enzymes to break it down if you don't even have it? That's a waste of time. It's a waste of resources. Don't do it. So what's going to happen? The repressor is always being made. What the repressor does is it goes and it binds right there between the promoter and the operator region of the lac operon. Now, if you remember from transcription that we just previously discussed, for transcription to occur, RNA polymerase has to bind to the promoter region. That helps to orient it and focus it and direct it in the right direction. When the repressor is bound between the promoter and the operator area, the RNA polymerase, number 1, it cannot bind to the promoter. Why? Because the repressor is taking up some of its space. It can't fit there. Even if it could fit there, it can't go anywhere because it's supposed to go from left to right, and you've got the repressor blocking it. It's like someone has just, excuse me, rudely, I'm blocking your way, and you can't go anywhere. If RNA polymerase cannot bind and can't move, transcription never occurs, which is okay because you don't have any lactose present. Now, what happens when lactose is present is shown in the figure B when the lac operon is induced. As I said earlier, you still have that repressor being made. It's made all the time. Now, when lactose is being made, what happens is it ends up binding to the repressor. If it binds to the repressor can't bind to the operator region, can it? It's basically removed it out of the way. So lactose binds to the repressor. The repressor cannot bind onto the DNA strand, so therefore RNA polymerase can bind to the repressor, to the promoter area. The repressor's not there, so it can bind, and then it can just pass right through over the operator region because that's no longer blocked, and it can start reading past those 3 genes, producing the mRNA that's needed now. Ultimately, afterwards, translation will occur, and you'll make the enzymes that are necessary to break down the lactose. Now, what's going to happen is that once you start making those enzymes and you start breaking down the lactose, how do you turn this off again? Well, eventually that lactose is going to be pulled off of the repressor, broken down, the repressor goes back and binds up to the operator region again. It's a very effective way of controlling the making of various proteins. Only make what you need when you need it. This is showing with the trip operon. I want you guys to be familiar with the lac operon. That was the first one discovered. That is the most well-studied one. But just know that different operons can operate in different fashions as to whether they're activated, whether they're repressed, whether they're induced. And the bottom line, it allows the control of a group of genes as a single unit, which allows you to adjust very quickly to changing environmental conditions. So this table is just showing inducible operons usually are regulating catabolic pathways, and repressible operons are usually regulating anabolic pathways. There are various types of RNA. I mean, we've talked about messenger RNA that's carrying the message from the DNA and then used as a template to make proteins. tRNAs are the carriers for the amino acids involved with protein synthesis. Ribosomal or rRNAs are actually part of a structural component of the ribosomes. Just be aware that there are also additional types of RNAs. So there's several different types of them that are used for various processes, usually involved with genetic expression. Mutations, this is where you have a change in that nucleotide base sequence. It actually is not that frequent of an occurrence. Sometimes it can be good, oftentimes the results are disastrous. There are different types of mutations. There are what we call point mutations and frameshift mutations. Point mutation is where just one base is affected. Oftentimes it's when you have a substitution. Frameshift mutations are where you have either added an additional base, so you've inserted things, or you've deleted a base. Either way, a frameshift mutation is called that because whether you delete or insert, it's going to change everything from that point on till the end of the protein. It will change it so that ultimately it's going to affect the codon sequence. And this is just a diagram showing some of the different types of mutations that may occur, where you have a substitution type of mutation. You're just changing one base. That may change the amino acid for that one spot. It may not. If that does change the amino acid for that one spot, that codon, so it changes one amino acid, depending on the location, it may or may not be disastrous. Frameshift, like I said, you can either insert or delete, and like I said, anything from that point on will change. So this shows types of mutations, the description, what some of the effects can be. How do mutations occur? Oftentimes it can be due to exposure to different things such as radiation, chemicals, etc. UV is a known mutagen. It causes what we call a thymine dimer. It changes where if you have two thymines next to each other in the DNA base sequence, they end up forming a bond between themselves instead of with the complementary strand, and you end up with this little bubbling structure here. There can be problems where you have replacement of the base with an analog, a similar shape, not the same shape, and that ultimately is going to have an effect on what the overall shape of the DNA is going to be. How frequently do mutations occur? Overall, they're relatively rare. During DNA replication, the DNA polymerase acts as a proofreader, so that usually it can pick up when it has put in the wrong base, backs up, cuts it out, puts the correct one in. It has been estimated that DNA polymerase during DNA replication is adding about 2,000 bases per second. So it's moving. There are different ways that the DNA can repair itself. There can be direct repair, single-strand repair, error-prone repair. This is showing some of the different mechanisms on how it can repair. How do you identify if a mutation has occurred? Mutants are those that cells that are descendants from a cell that had a mutation that was not repaired. Wild type is what's normally found in nature. You can have different ways of trying to recognize or identify mutations. Sometimes mutations occur where, say, originally you had a group of bacteria that were sensitive to penicillin, and then a mutation is where one becomes resistant to it. Well, those that are sensitive, if you grow everything on a medium without penicillin, everyone should grow. If you then have a second medium where you add penicillin, only the mutant that is now resistant will be able to grow on there. So you can identify it and you match it up. And then you isolate from that one. Sometimes you can add a particular growth requirement that is necessary for the mutant to has to have in order to grow. And so you can inoculate media and you use a stamp from one to the other, one that has the medium, one that does not have, or one that has the special growth factor and one that does not, and you compare once again. Sometimes you can add a dye, so you look for a certain color. So there are different ways that you can look, it just depends on what you're working with and what you are testing for as to how you can identify the mutant. Years ago when I was in graduate school, I had a class, we were doing a genetic engineering class, working with a professor. The way we were identifying the mutants, she was working with what's known as ice minus mutants. Certain bacteria that would lower the freezing temperature from 32 degrees C to 32 degrees Fahrenheit to 28 degrees Fahrenheit. The way we were looking for mutants that had this gene that would lower the freezing temperature. So the way we detected them was we would grow all of the different cultures in separate test tubes in a liquid broth. Then we actually would take a water bath that we would put ice in, drop the temperature down to about 30 degrees. And then we would just literally line a sheet of aluminum foil, and one by one we would, in rows, just drop a drop of our samples. If it froze immediately, we didn't look at it. Those that did not freeze, we knew were the mutated ones. Oftentimes in nature, there is exchange of some genetic material. Recombinants are cells with DNA molecules that have a new sequence of DNA in them. This is showing a diagram with genetic recombination. Up at the very top, you have got a DNA strand labeled as A, and then you have another strand labeled as B. As you can see, an enzyme comes in, cuts strand A, and then part of that DNA comes in and exchanges with an area that has also been cut on B. And so a piece of A goes to B, a piece of B goes to A, so now both of them are recombinant. They just have something in them that was not there to begin with. With bacteria, one of the things that we have looked at is transfer of DNA. How does this occur naturally in the environment? And there's 3 different ways amongst prokaryotes that you can have what we call horizontal transfer from one organism to another. And the 3 ways are transformation, transduction, and conjugation. Transformation is essentially when you have a little piece of DNA that is taken up, usually from the environment. It's often referred to as naked DNA. And so this is showing different ways how they have studied this in the lab. But basically, you have naked DNA from the environment that's taken up by the cell. Now, they were studying this with bacteria and then using it with mice, injecting the mice with it. Transduction, in this case, what you're using is a virus. A phage is a virus. And some of that viral DNA is transferred and taken up by the recipient. So in this case, you have at the top the virus coming in, it's injecting its DNA into the cell. Now, normally it's going to go through. That DNA starts to replicate, the viral DNA starts to replicate, making new viral particles. However, usually the bacterial DNA is going to be destroyed. What happens in this particular case, it has taken up some of the DNA from that host bacteria, and the new virus that has that bacterial DNA goes into infects a new host cell, and some of that DNA gets incorporated into the host DNA. So transformation, you're taking DNA from the environment without help of any other like viruses. Transduction is with the virus transferring the DNA. And then the third type is conjugation. And this is where it involves the pili, that small connection that some bacteria form that connects one cell to another. It's kind of like a hollow tube, and you can transfer material from one cell to the other. Oftentimes this is how plasmid, when a plasmid goes through its own replication, it goes through replication on a different schedule than the main DNA, and it can replicate and be passed and transferred from one cell to another. Now, the two cells must be compatible. Usually it's only gram-negatives that will form this pili, which in the upper top photo of A, you can see a really neat picture showing the two cells with the actual pili connection. It's only with gram-negatives, and even though it's not all gram-negatives, they have to, number one, be the same species, and even if they're the same species, they must be compatible in order for this to work, and they must be connected long enough that the entire plasmid does pass over. Why should you be concerned about this process of conjugation and the transfer of plasmids is because on the plasmid is often where there is the resistance genes to antibiotics. So this is how antibiotic resistance is often transferred. So the original cell, cell 1, with that plasmid may have been resistant to, let's say, erythromycin. And cell B was susceptible. Well, by the time it gets past at the very bottom, now cell 2 also has resistance. So there's just different ways that this can occur as seen here. Just know, once again, the differences between those three methods of transferring DNA that increases diversity, genetic diversity for prokaryotes. Transposons are simply just little segments of DNA that can move from one location to another, either on the same molecule or different molecules. They can basically flip, and this is just showing that the transposons can move from one area of the DNA to another. They can replicate, and then they can move into plasmids as well. Insertion sequences are the simplest of the transposons, but some of them become more complex. So it's just showing how they can flip.